Hey everyone, here's a little video to talk about different microscopes. Since we're talking about cells, you can't see cells with your bare eye. You have to use microscopes. And there's different types of microscopes that you might use to see a cell. So here they are. The first one is a light microscope. They magnify small specimens that you can't see with your naked eye or bare eye. They use a beam of light. It actually comes out from this part, which is the light source, comes through the stage and through the objective lens, through the body tube, and to the eyepiece or ocular lens. And that produces a 2D image of a specimen for you to see. Now, with our microscopes, our light microscopes, the maximum magnification is oftentimes about 400 times magnification in our classrooms. You can have some light microscopes that'll do 1,000 times magnification, but not in our classroom. So there are some different parts of this light microscope that are important to note. There's the base that it sits on and the arm. And if you're carrying a light microscope, you want to hold the base and the arm. Then there is a light source and a diaphragm that controls how much light is actually coming through the stage to your eye. And this is called the stage. And that's where you put your microscope slide. You can hold the slide in place with stage clips, and you can use either low, medium, or high powered objective lenses, and they're on what we call a revolving nose piece. So that can actually turn and help you choose which type of lens you would like to use. That is attached to the body tube, and that is attached to what we call either the eyepiece or the ocular lens. And this is also something to note here. There are two adjustments, and those two adjustments, the fine adjustment and the coarse adjustment, move the stage up and down to bring the object into focus that you want to see. Well, the coarse adjustment moves the stage a lot, and the fine adjustment just moves it a little bit. So when you're first focusing in on an object, you want to use the coarse adjustment. But once you've focused and found it, and you switch to higher power lenses, you're only going to use the fine focus after that. Next up, we have scanning electron microscope. Now, this microscope is used for items and specimens that cannot be seen with a light microscope. They're even smaller. You can use it for things you can see with a light microscope, but it can even see things that light microscopes are not able to see. They don't use a beam of light, but instead use a beam of electrons and produce 3D images of specimens. And they can magnify things up to 1,000,000 times. As you can see, they're pretty large. They take up actually a small room rather than just a small space on a lab station. So here they are side by side, the light microscope, not super complicated compared to the electron microscope, which involves computers and takes up a lot of space. So big differences. Light microscope uses a beam of light, while scanning electron microscope uses a beam of electrons. The light microscope is a relatively low magnification, while the electron microscope is high. Light microscopes are easy to use, and scanning electron microscopes are complicated. Light microscopes are inexpensive, and scanning electron microscopes are expensive. For light microscopes, you can see things that are either living or dead, whereas scanning electron microscope, you can only see dead things because the electron beam actually kills specimens. So even if it was alive, it won't be anymore if you use a scanning electron microscope. For the light microscope, the lenses are made of glass, while the electron microscopes are made of electromagnets. From light microscope, it's images formed by the absorption of light onto your eyeball, the retina of your eyeball, whereas the electron microscope forms images by scattering electrons, and those are brought up on a computer screen. The light microscope can produce color images, while the scanning electron microscope is only black and white. The light microscope is high resolution, and the electron microscope is light microscope is low resolution, while scanning electron microscope is high resolution, and light microscope is 2D images, while electron microscope is 3D images. Here's again kind of how they work. So in the electron microscope, you have a specimen and a beam of electrons is hit onto that, and it ends up collecting data on an electron detector because it scatters the electrons, and then the image is produced on a computer and your eye can see it. But on a light microscope, it's much simpler. There's a light source. It's shined through a specimen and then projected directly onto your eye. Here is a difference in some images produced by the different types of microscopes. This image is produced by a compound light microscope. You can see it's 2D, it's very simple, it's not very high magnification or high resolution. This one is produced by an electron microscope, and you can see that it's three-dimensional image with very high resolution and could become even much higher magnification than it is shown here. Thanks so much for listening.